Hello guys, welcome back to Gaurav Tutorials. In this video, we will be talking about how is the peptide formed in the structure of the protein. So, if you ask yourself a question that what is a protein, you would get an understanding that a protein is a long chain of amino acid held together by bonds. Okay, so this long chain of the amino acid that you see can be called as a protein. Okay, or it could be also referred to as a polypeptide chain. Now, since this protein is a polypeptide chain, I mean it's a polymer. What is it? It's a polymer, right? A polymer of amino acids. The polymer of amino acids. Depending on what these amino acids are, if you have the long chain of amino acids to form a protein and all the amino acids are same in the chain, then the protein will be called as a homopolymer. Okay, it will be called as a homopolymer. However, that is not the case. You will never have a protein which will have all the amino acids same in its structure. At least for time being, we do not have such protein which is a homopolymer in nature. Then if all the amino acids, like if the amino acids are different in the chain, then you call such a protein as a heteropolymer, or you can also call it as a copolymer. Okay, all right, so all the proteins that you see around you are heteropolymer. Okay, here the monomer in case of the protein would always be a amino acid. Okay, the monomer of the protein would always be amino acid, and when these two amino acids come together, when two different types of amino acid come together, they form something called as dipeptide. They form something called as dipeptide, and the bond formed between these two amino acids will be called as a peptide bond. Okay, you understand that when two different amino acids or same amino acids come together by forming a bond between them, they form a dipeptide, and whenever these bonds are formed between the amino acids, the bond will be referred to as the peptide bond. So our major focus in this video will go on how are these peptide bonds formed. For that, I'll take you to the skeletal formula for the amino acid. It goes this way. You have got NH2, there is R, there is COOH, and then there is H. Okay, now what am I going to do is, for the sake of understanding, I can rearrange these functional groups to form a structure like this. You've got carbon, you've got N, you have a H here, you have a H here, then there goes your R, the alkyl group, you have got C, double bond O, and then you got O, and you have got H, and one of the H with the C here. Okay, so boys and girls, I need you to understand one thing. You find these two structures to be different, but that is not the case. This, when observed properly, would give you an understanding that it's a carboxyl group, and when this is observed, it will give you an understanding that it's nothing but NH2 group, the amino group. Okay, so what will I do now, or what is going to happen when these amino acids, let me decrease the size and get a copy of this. Yeah, so you got these two amino acids coming together, aligning together. What will happen? One of the amino acid will find that they will be, I mean these two amino acids will find that there will be hydrolysis taking place. Okay, there will be hydrolysis of the peptide, these two amino acids would take place, and they would give rise to, they will form water, and in turn, as soon as the water is removed, they'll form a bond between this carbon and N, the C and N. Okay, so this bond that is formed by the removal of the water molecule between the two amino acid is called as a peptide bond. Okay. Similarly, what you will find. So let's drag it here. Similarly, you'll find the water molecule formed by donation, removal of this OH from the carboxyl group and H from the amino group. Okay, this OH from the carboxyl group and H from the amino group would be left out and forming a water molecule, and the bond between this carbon and the carbon of the carboxylic group and N of the amino group would again be referred to as the peptide bond. Okay, so that's a peptide bond. Now, if I actually zoom this thing out, I would want you to understand, okay, here for this amino acid, the new bond, if at all is to be formed, will be formed on this carbon. So this free carboxylic group is termed as C prime. For this particular amino acid, the new bond will be formed on the amino group. So this N, the amino group being free and available for the formation of the bond, will be called as N prime. Okay, so you will come across the structure of the proteins whereby you will be having a helix and you will find there is N prime and then there is C prime. So we need not worry what is this N prime and what is this C prime. Whenever you see this N prime, you get an understanding that if at all a new peptide is to be added or if a new amino acid is to be added, this amino acid would be added to the amino group. When you see the C prime, you'll get an understanding that if at all the amino acid is to be added, it will get added to what functional group of the amino acid? It will get added to the carboxylic group. All right. So you have got the right side at the right side of the amino acid, okay, with N prime, and left side of the amino acid with C prime. So that is it, guys, in this video. I hope you would have understood the video. If that is the case, you can hit the like button. You can also share the video with your friends, and if you find the channel to be helpful, you can also subscribe to the channel. See you in the next video. Till then, take a very good care of yourself, and yeah, God bless you.